Mr. Mark Bale, Chairperson of the Council of Europe Committee on Bioethics, and Mr. Luigi Naldini, Director of Division of Regenerative Medicine and Gene Therapy. Welcome to Mediabox, both of you. And I would like to first start with you, Mr. Mark Bale, because you participated in a meeting hearing on manufacturing and new human species in the Parliamentary Assembly session today. So it's an interesting topic, interesting subject as well. What should we understand from the manufacturing a new human species? What's that exactly?
Okay, so I think it's a prospect that's been discussed for a long time. As I said, it's an issue that the Oviedo Convention, the Convention on Human Rights and Biomedicine, anticipated in the 1990s. I think what's started to happen, from my understanding of this, is the techniques are now much easier to do and they're much more precise, and they begin to become something which is therapeutically useful. And so we have to really think about whether the requirements to protect and nurture the kind of embryo and to only use these techniques of germline editing for preventative and therapeutic methods or reasons rather than enhancement. We have to get that much more fully understood. We have to have that debate in the democratic society, and we have to think about guidelines and sort of rules for scientists and others to understand.
Mm-hmm. And also in that point in the meeting, Mr. Naldini, you mentioned about gene therapy. So what is gene therapy exactly, and how can it be more efficient that you mentioned in the meeting?
So gene therapy means using gene as drug, essentially, rather than delivering a small molecule, delivering a whole gene to a patient's cell to treat a disease. Mm-hmm. This usually can reverse or correct a genetic mutation by adding a functional gene. Mm-hmm. This is what is happening in the clinical trial ongoing, and seems a promising strategy to treat a variety of disease. Mm-hmm. But this is using body cells, somatic cells. So this treatment will only affect the patient and will essentially end with the patient. Now there are new techniques which we are very excited about, which are called gene editing technology, mm-hmm. by which rather than adding genes, we can rewrite the gene. So if there is a mutation, we can correct that mutation and bring that gene back to a normal function. So this is really expand the scope and the precision of what we can do, but these are early days for this technology. We are experimenting them on body cell, but there is a possibility of using them also on germline cell, potentially to avoid to pass on a mutant genes to the progeny. But that is also ask issues, how safe is to touch the germline, how ethical is acceptable. And that's where I think we need to have a debate and have a consensus how to move forward.
Mm-hmm. And these kind of debates always remind us a question of ethics. So ethic and human rights perspective and limits of this ethic. So what can you say from that point with this new technology? Do we need to discuss, or is it really impact of a problem of ethic or limits of ethic?
Yes, I think it's a big ethical debate, as we've heard today. I think there are lots and lots of interesting perspectives about when you might see this as being something which is the only hope that a parent might have to have a child unaffected by disease, mm-hmm. right through to those who think there's a fundamental kind of religious reason why we should not be doing these things, and that's a spectrum that we have to reflect on. I think that there are some very important if you like, lines that we should probably be laying down now. And these are already set out in the Oviedo Convention, but I think we should be clearer what they really mean. And that's really to say that we can, I think, use techniques like gene editing to do some very important, powerful therapies, as Professor Naldini says. We can also use them to understand what goes wrong in the early embryo, in early miscarriages in pregnancy. And that should probably be highly supported. But beyond that, I think it's far too early to say what we should be doing with these techniques, and we should be having a debate about that, an ethical debate and a democratic debate.
Yeah, and as a human being, generally, when we talk about new techniques, we always ask, so how can it influence human beings? So what can we have with this new technology? What can you say about that point, Mr.
Well, I think we can have a lot in terms of the possibility to intervene on the genome and changing the inherited gene which is defective. So that's quite an amazing result, and I think that's where we are now in terms of trying to get the benefit of this new technology. And moving within the framework of treating disease and experimenting this strategy, proving how they're safe. This may extend, as mentioned, to potentially preventing to transmit a disease gene. But then we are really entering an area which, as mentioned, has to be carefully thought before we really move in.
Mr. Bale, Mr. Naldini, thank you for joining us and for your all contributions. Thank you.
Good.
Thank you very much.